Boa noite, sejam todos muito bem-vindos. Estamos iniciando o nosso quarto workshop do Programa de Ciências Farmacêuticas da Universidade Federal do Espírito Santo. Eu sou Lorena, pós-doutoranda do programa, e o objetivo do nosso workshop é apresentar nossas linhas de pesquisa para os programas, de mestrado e doutorado. O nosso workshop se inicia hoje, com a palestra do professor Eduardo, e continua na próxima segunda-feira, dia 27, a partir das 8 horas da manhã, no prédio do nosso programa, Uma informação importante a todos presentes: teremos um formulário de presença, e esse formulário ele vai contar com uma palavra-chave que será apresentada durante a apresentação do professor Eduardo. Então, fiquem atentos que vocês precisam preencher essa palavra-chave para emissão do certificado de participação. Um bom workshop a todos. E agora eu passo a palavra para o professor Rodrigo Kitagawa, que é professor e coordenador do nosso programa de Ciências Farmacêuticas. Boa noite, professor.
Boa noite, Lorena. Boa noite a todos. Com a imensa satisfação que nós vamos iniciar esse quarto workshop do programa de pós-graduação em Ciências Farmacêuticas. E o início desse workshop vai ser com a palestra do professor Eduardo Caro, que é da Universidade de Porto Rico, onde o professor ele vai falar sobre produtos naturais marinhos, que é a área de expertise, que ele atua na universidade. Como a Lorena falou, nós vamos ter algumas atividades aí a partir de segunda-feira, com a visita dos laboratórios e também para o conhecimento, eu vou apresentar o edital de seleção de mestrado e também os professores vinculados ao programa, assim como suas linhas de pesquisa. Nesse momento, então, eu vou apresentar o professor Eduardo Caro. First of all, I would like to thank Professor Eduardo Caro to accepting our invitation to talk about marine natural products, which is his area of expertise. Eduardo, would you mind introduce you in Portuguese? That's okay?
Yes.
Ok. Então, o professor Eduardo, ele é bacharel em Química, graduado em 2008 pela Universidade de Porto Rico, fez pós-graduação na Universidade da Califórnia, em San Diego, onde trabalhou com síntese total de produtos naturais bioativos, defendendo seu doutorado em Química Orgânica em 2014. Em 2016, ele ingressou no Scripps Institution of Oceanography, onde trabalhou no Centro de Biotecnologia, Biomedicina Marinha, sob supervisão do professor Dr. William Garrick, estudando isolamento, síntese e caracterização biológica de produtos naturais de cianobactérias, sendo bolsista de pós-doutorado do NIH Cancer Training Grant. Em 2019, se tornou professor de Química Medicinal na Escola de Farmácia da Universidade de Porto Rico, no campus de Ciências Médicas, onde atualmente desenvolve pesquisas em descoberta de novos compostos a partir de produtos naturais marinhos. Em outubro de 2023, o professor Eduardo esteve em Vitória e ministrou uma disciplina nos programas de pós-graduação em Ciências Farmacêuticas e pós-graduação em Química sobre produtos naturais marinhos, além de coletar algumas amostras, firmar parcerias em projetos em desenvolvimento. Então, thanks again, Eduardo. So, feel free to start your lecture.
Boa noite a todos. Thank you, Rodrigo, for that introduction. I promise next time I give a talk, my Portuguese is going to be so good that I will give the whole lecture in Portuguese. So I have to keep practicing. So I'm originally from Puerto Rico and did a lot of my graduate work and research outside of Puerto Rico, but I moved back in 2018. and now have been 6 years at University of Puerto Rico, now as Associate Professor of Medicinal Chemistry, where I started a research group around marine natural products. And as you heard from my background, my training has been in organic chemistry, synthesis, medicinal chemistry, and now we do some of the natural products chemistry. And I decided to combine all of those into one research program. Today, I didn't know where everyone was going to be at in terms of knowledge. So let's see if I can get my presentation going. If it wants to let me. So I wanted to start with what pharmacognosy is. It's very probable that you have all heard this concept. Maybe you have taken classes and it's part of your coursework. I think this term is now has evolved. And so the original term derives from two Greek words, pharmakon, which means pharmako, right, drug, and gnosis, that comes from knowledge. And now I think a lot of us think of this not only as the use of natural substances, biological substances, as medicine, but I think now pharmacognosy really aims at studying natural product molecules. So it is a field about chemistry, right? These are also known as secondary metabolites. We'll talk a little bit about that in a second. And apart from their medicinal properties, they also have very important and prominent ecological functions, which is the origin probably from their bioactivity. And also they have found their way into the food industry and, let's say, other industries. We know that probably one of the most important antiparasitics comes from a natural product for cattle and different kind of animals, right? And so the veterinary industry has also benefited quite a lot from natural products. And I think traditionally, probably you all have been studying natural products from plants because plants are a big producers of natural products. But I think now the field of pharmacognosy really spans all biological kingdoms, and we include here fungi, bacteria, especially have become very popular because of their the ability for us to control cultures in the laboratory. And so we can grow fungi and bacteria reproducibly. We can scale these up, and the biotechnological implications of producing compounds based on these. And the other part is, maybe I'll talk a little bit today, is about marine environments, especially marine invertebrates, which now we understand that most of the diversity that comes from these natural products is also somehow of microbial origin. And so based on what we know now of pharmacognosy, I don't think we can talk only about chemistry or phytochemistry or botany, but marine biology, microbiology. Now that these compounds have worked their way into the clinic as single chemical entities, we can think about pharmacology and how these are produced on big scale, so biotechnological applications, and also how do these apply for those who are more interested in the clinical applications. So pharmacognosy itself, I think, is very highly interdisciplinary, very rich in terms of the fields that you interact with. And if you were to go to any natural products or pharmacognosy conference, you would probably interact with a diverse field of scientists, including marine biologists, pharmacists, chemists, and microbiologists. And so I think that's a really neat and unique aspect of natural product research. So I am part of the American Society of Pharmacognosy, not the only society. There are many other pharmacognosy societies. And this is their most or let's say the most updated definition of pharmacognosy, which is the study of natural product molecules for their medicinal, ecological, and gustatory and functional properties. They span all these kingdoms. And I think the most important part I wanted to share with everybody today is it's ever changing. And I think now we've taken a lot of inputs, especially from the updates and advancements in genomics, where we see a lot of people who are discovering natural products from that standpoint, and other omics fields. But constantly these technologies can feed natural product research. And as they're developed, I expect to continue to see changes in directions of how we approach the discovery and development of natural products. And so We can date some of the natural products. These are some classic stories. Maybe you've heard of them. I won't take too much time, but the white willow tree used for fever, dated for at least 400 years before Christ, used as in tea form, later on seen to produce salicylic acid. Discorea villosa, and this is one of my favorite stories, is the use of these wild yams, which was used in complementary ways, meaning that it was used in toxic levels to act as a birth control, but also a tea of the root or a different part of the plant would ease contractions during labor. So a lot of indigenous populations had an abundance of information of how to use these resources medicinally in a very effective manner. And also, I think importantly for all of those who are interested in this is kind of the basic introduction I give to most people about natural products. Is it's a chemistry field. And so for those that are out there that are interested in doing chemical research, natural products at the most fundamental level is based in organic chemistry. And this comes from some really important discoveries in the 1950s by Rule, who isolated urea, and then Scheel, who isolated some organic acids from citric fruit. And finally, probably the most important discovery, which is the discovery of morphine from opium, which was very much the first single chemical entity we had derived from an extract, right? And to this day, still morphine plays a really important role. And actually, most of the discovery of opiates are derived from the structure-activity relationship or structural modeling of morphine. And finally, I think when synthetic chemistry was discovered, maybe not such an interesting molecule to us now, but then to understand that we can go from inorganics to organics, and in general use chemical transformations in the laboratory to produce carbon-rich chemicals, was a huge advancement in the understanding of organic chemistry and really set up the stage for many discoveries to come. And so this kind of entered the era of single chemical entities, and you've probably heard this many times, but apart from using natural medicines in their extract form or in, let's say, their complex form, we started to understand that, especially from the discovery of morphine from opium, that there was certain molecules that were very much driving the bioactivity and the pharmacological activity of these extracts. And so many of these you're going to see: strychnine, quinine, caffeine, nicotine, cocaine, atropine, all alkaloids. There's very specific chemical ways of isolating these compounds that really led the way of why these were some of the first compounds. But in general, really important compounds, especially here, nicotine described later on. Most of the nicotinic receptors and then muscarinic receptors as well were discovered because of nicotine. Atropine as well, right, as an antagonist of or an anticholinergic agent, and some stimulants in caffeine and quinine, some really important molecules. I think caffeine is something most of us consume on a daily basis. And so if we were going to define a natural product, instead of naming it a chemical compound or substance produced by a living organism, which I think is too generic, I think for today's talk and in general moving forward, if we're going to think about natural products for drug discovery, then we really need to refocus that and put in a second definition, which is these molecules that are produced via the secondary metabolism of living organism. And the second part is especially important, that they have highly specialized biological function. And I'll talk a little bit about why that's so important in terms of drug discovery. So as a reminder, maybe you've already seen this, so I'll continue to move pretty quickly. In terms of primary metabolism, we think about those chemicals and compounds in the body that are necessary for life, so development, growth, reproduction. It's shared among all living organisms, so we can think about urea, which was one of those really early discoveries, and lactic acid and amino acids, which are basic for the formation of proteins. But when we look at secondary, these do not give a direct or are not involved in direct metabolism, meaning that they are not required for the basic functions of life, but they are highly specialized for some kind of function that provides either an ecological or evolutionary advantage to the organism. So for example, many of us don't recognize this, but as humans, we produce antibiotics. These antibiotics are actually quite complex in structure. And maybe if you get a cut or you have some kind of opening and you can see that it's been infected, you can probably isolate some antibiotics from your own body from your secondary metabolism. But we also see like morphine, steroids, flavonoids. I can think also about cannabinoids, right? Which even though they have found a function in our central nervous system, are really there for other purposes, which provide advantage in terms of reproduction to the cannabis plant. So in general, again, primary metabolism, think about fatty acids, carbohydrate, nucleobases, things that build these really basic building blocks of life. And when we think about secondary metabolism, we can already go straight into the ocean. And so here are some, one of many conotoxins from Conus magus. This is a cone snail derived from the snail, right? It has like a nice cone if you extended the picture this way, and a snail that leaves underneath. And this provides a very highly tailored function. And I usually play a video, and it might be here in my slides, but if it's not, I might go forward because we have a good amount of ground to cover. But basically, this has a proboscis that comes out from this tube, and it has a harpoon loaded with these conotoxins, over 100 of them, that then immobilize its prey. And in this case, since the snail is quite slow, the advantage it gains from producing these kind of secondary metabolites is for those, for it to hunt efficiently without spending too much energy. And so, as I said, if you're interested, you can find this video by National Geographic. It's quite dramatic. You can look for hunting cone snails. I would be more than happy to share a link with everybody afterwards. But in the interest of time, I will continue to say that after much trials and tribulations and work from scientists, in 2004, the FDA approves the conatide or Prialt, one of the many conotoxins, so a single chemical entity derived from this venom, as an analgesic agent, mostly because of its interaction with N-type voltage-gated calcium channels. And in this case, blocking those calcium channels brings analgesia and basically disrupts the central nervous system from perceiving pain. And the other part I want to discuss in this is that obviously we can repurpose these natural products for drug discovery, but there's a big role in chemical ecology now. And for those of you that are maybe interested in moving into the field of natural products, this to me is one of the fastest growing fields, which is what is our basic understanding of what these natural products are made to do in nature. And we can learn a lot from the chemical ecology, and a lot of this chemical ecology can actually translate pretty readily into pharmacology. We know that the proteins in our bodies known as receptors that interact with these molecules are probably homologs of other kinds of receptors that are found in our natural environment. And so it's very possible that if you understand the chemical ecology of these compounds, you can then translate that into developing it as a pharmaceutical. And in the case of plants, you can think of these as compounds that are produced in defense, especially against herbivores, and also against microbial pathogens. We also have quorum sensing, which is basically how bacteria communicate. For a very long time, we thought that bacteria were at war with each other. And I think a lot of new research has shown that, in fact, it's not necessarily warfare. It's not that they're creating really cytotoxic molecules to kill each other off, but that these molecules communicate or disable certain functions that allow them to somehow live in certain spaces without getting on each other's nerves, if you would see it that way. And then I think there's some other understandings of allelochemicals, or we've seen these effects in the environment. I'll provide a couple examples, specifically here in the Pacific Ocean and here in the Caribbean, of those compounds that are secreted, and these are mostly done through microalgae in the environment, and somehow affect growth and development of mostly mammals, or somehow find their way into the food chain. And there is some research, but it's still not completely understood the why these compounds are being produced, and still some interesting questions to be answered there. So one example of these allelochemicals that I can provide is a great story that came from California in the shape of a small molecule called domoic acid, which produces amnesic shellfish poisoning, also known as ASP. And so here you see a sea lion. And what many people were observing was these mammals were actually suffering seizures. You can see some foam coming out the mouth. And this was because they had been intoxicated either through the food chain or directly through domoic acid, through the Pseudonecia microalgae that produces the toxin. And then it can affect humans as well, in terms that mostly shellfish will integrate this compound through the food chain, and they can retain the toxin because they do not have an ability to metabolize it. And so in this case, that toxin can move through the human population if we consume yummy seafood. I am a big seafood fan, especially living in the Caribbean. And this will produce permanent short-term memory loss in severe cases of ASP. So here's one understanding of a full story between a small molecule produced by a macroalgae integrated into the food chain. And there's understanding and genetic understanding of why this is produced and certain things about how it's triggered. This is work that was done at SIO at UCSD. But I think still there's a lot of questions of exactly why is this necessary in the environment. Since I'm from the Caribbean, we also have certain other interesting phenomenons happening here. These are polyethers known as ciguatoxins, produce ciguatera. And I don't know if you've heard about ciguatera, but here there's a news story from 2020 where 21 persons were intoxicated with ciguatera poisoning by consuming picua, which picua is the same thing as barracuda. And so many coral reef fish collect this poison from the food chain. These usually big reef fish are unable to process these polyethers. As you can imagine, maybe some of you have understood a little bit about metabolism of compounds. These are very hard to metabolize. There's not many groups to be oxidized by our phase 1 and phase 2 metabolism, so they can circulate in our bloodstream for a very long time. And it produces quite disgusting and unpleasant symptoms, from like weird sensations of temperature and taste, all the way to amnesia. And so this is now known that not only the ciguatoxin, but the brevetoxins and other toxins are interacting with certain voltage-gated sodium channels. And those targets are kind of well understood at this point. And so again, here's a story about a compound made by a microalgae that moves through the ecosystem and somehow can affect humans indirectly. And so if we understand what these are for, maybe there's some homologs of these, let's say for pain, we can see that there's other voltage-gated sodium channels here for the codotoxins, but some of these others can be very harmful. Okay, and I could continue down this line, but in general, what we want to understand is that the genetics of these organisms, these algae, these plants, these invertebrates, code with the genome, and those genomes have a certain part or fragment of them dedicated to producing natural products known as biosynthetic gene clusters. And then those biosynthetic gene clusters at their core are proteins that perform some kind of enzymatic transformation. And then if you align them all together, you can produce then these natural products. And so I think this is really important because we can see a clear relation of how these compounds are of genetic origin. So it makes sense that these compounds somehow would interact with some kind of protein target. And so I'll say something kind of provocative here, and that's that natural products are made to fit. And I will make one really big disclaimer, and that is not that these compounds are designed with us in mind. I don't think nature cares that they're making these molecules and they're thinking of us when they're making them. I definitely think that they're making them for their own ecological purposes and their ecological advantage. But we can definitely believe that if the compounds are made so specifically by these biosynthetic gene clusters coming from these genetic cassettes, that they're made to interact with another receptor, and that those receptors that are in nature somehow can find some homologs in ourselves, and there has to be some kind of association there. And so I would very much think that these compounds are designed to interact with proteins and receptors, just not ours. And we can then definitely repurpose these for drugs and drug discovery. And so I can talk here for probably another 20 minutes or 30 minutes or maybe like 3 hours of natural product stories. We can talk about cannabis and its producing of these really important cannabidiol and tetrahydrocannabinol, THC, one that has been used as an appetite stimulant and anti-nausea, failed miserably in the 90s as Marinol, but it's found its way back as an extract from the flower of different kinds of cannabis species. And most recently, CBD found its way for the treatment of seizures, especially in young adults and children, and in the form of a nasal spray called Epidiolex. Probably one of the most famous stories in natural products is the discovery of Taxus baccata after the discovery of Taxol. This is because Taxus baccata produces diacetylbactam-3, and this semi-synthetically can produce Taxol, the most sold and used anti-cancer drug of all time. We have our microbe friend Streptomyces. Streptomyces cattleya produces one of the most potent antibiotics in dianomycin. We have our other microbe friends, the fungus among us, in Trichoderma, who can produce immunosuppressive agents such as cyclosporin A. And obviously, we talked about the ocean environment, and we talked today about saconitide. And so this is just a brief synopsis of what natural products are and what all the indications these can have. And I'm still very excited about this field because I feel like it's had a renaissance in the last couple of years where people are getting increasingly more excited to research these, given the new tools we have to look into them. And then Puerto Rico definitely has a story that's, I think, undertold, which is the discovery of tribectedin, or ET743. I'm actually quite proud of this picture that I took at Hobos Bay in a quite clear day of this beautiful organism, which is still one of my favorites to locate in nature when we go snorkeling here, which is Actinicidea turbinate. It's also known as the mangrove tunicate. And in the 70s and 80s, this tunicate was harvested and extracted for its anti-tumor properties. And it wasn't until 1990 that Wright and Hart and co-workers were able to establish the structure of one of the major compounds, tribectedin ET743, which is now known as one of the most, or the first unaltered marine natural product drugs. And so again, I kind of summarized it here. First discovered in the late 60s, developed in the 90s. PharmaMar, a company in Spain, picked up this project and was able to biotechnologically produce a microbe, and they branded this Yondelis tribectedin and indicated its use for soft tissue sarcoma and relapsed ovarian cancer. And then again, we talked about chemical ecology and its importance. It turns out that the tunicate itself doesn't really make the compound. It's an endosymbiont that lives inside the tunicate. These tunicates are filter feeders, so water comes out and in and out all day for them to feed, and in that process, bacteria get accumulated in these tubules, and it seems one of the endosymbionts was producing this. And as soon as they were able to figure this out, they were able to find another bacteria that produces cyanosasafrin C, which provides most of the difficult stereocenters and cycles for this molecule, and then semi-synthetically acts as tribectedin, or ET743. So again, I think chemical ecology, not only from the standpoint of understanding the pharmacology, but also the biotechnological implications it has, is very important in natural product research. Most recently, SARS-CoV-2, we have some tunicates. I actually have been looking at the literature. There have been some clinical trials now for plitidepsin, which is made by, or originally isolated from another tunicate, Aplidium albicans. It's quite likely that this is also of microbial origin based on the structure. It's not exactly clear, but there has been some reports of some of the microbes thought to make these. And I think there's already some reports of exactly some microbes producing these plitidepsins. And so I'm excited to see where this story ends up if we have some new protease inhibitors, especially this host protein EE-F1A, as a new target and therapeutic option for the treatment of COVID. And so with that, I've kind of given everybody, hopefully everybody's on the same playing field. I will move quite quickly because I think I usually take up too much time. I talk too much. But my lab, as I said, started in 2018. And my lab is very interested in the center part of this Venn diagram, which is new structure-function relationships. The way we can approach this is in different ways. So for example, I talked about marine natural products. So we do go in the ocean, we collect, we extract. We're very interested in new natural products, structures. We're interested in new bioactivities, that be from new structures or known structures. And we also are very interested in using metabolomics as our main tool to explore those. And so in that corner, we have an antivirals program. Some of these students have actually moved on now. Gracie is at University of Florida. Victoria graduated recently last year, now is at Caltech doing her PhD. And Chris is at the School of Pharmacy at UPR doing his PharmD degree. Marie Matos is actually almost on her way out as a fourth-year graduate student. And we recently published a paper in the Journal of Natural Products showing our work on SARS-CoV-2, and I'll talk about that today. We also look at anxiolytics, and this is a project that's still ongoing. Laura has also moved on as of right now, but we're using a fly model to see if we can find some new compounds to keep everybody chill. As of right now, we're just trying to figure out how these brown algae, macroalgae, are inducing a reduced effect of centrophobism, which is a measure of possible anxiolytic activity. We're not interfering with the fly's motor capacity, but we are trying to understand if whatever effect is happening in the fly has a proteomic profile that we can understand what's happening in terms of the signaling to isolate maybe some new anxiolytic compounds. We also have some, and I actually failed to mention, we have a couple of new members. My lab has changed quite a bit in the last 6 months, but Alexandra, Juliet, and Nayar have all moved on. They've been working on another natural product, Majesticamide D, and this is from our synthesis portion where we try to study compounds through synthesis and variation of the compound to understand structure-activity relationship. And finally, I'll be talking a little bit about our pseudo-natural products project today at the very end, where we're trying to find new ways of just designing from scratch between synthesis and medchem, new compounds completely based on structures that are published in the literature. And so this is a nice picture for those of you who want to get excited about this. These are all pictures of things we've collected in the last 6 to 7 years. Maybe some of these you recognize. There's some macroalgae here known as the sea lettuce. This is ova species of green algae. We have padina, which is a brown algae, which we're looking for anxiolytic properties. We can see some things that look like filamentous varieties of micro and macroalgae. Here's some macroalgae. Here we have some filamentous diatoms. But also you're going to see a good amount of these are of cyanobacterial origin. So 1, 2, 3, 4, 5, 6 that I can understand. I'm excited to say that we are just isolated a new compound from this guy right here. We're still trying to identify the species of this algae, but we can see different colors, textures, shapes, depths, and ecological niches where these grow. So this is why we like algae. They're easy to collect. We get a wide diversity of compounds. And I think they're slightly understudied. I think a lot of people studied these a long time ago with the tools that were available. And we have other tools now, such as metabolomics and better analytical techniques to dive a little bit deeper into them. So we had one problem, and that was COVID. I think that was for everyone. We had probably one of the hardest shutdowns across America, to the point where our lab was closed for at least 2 to 3 months. And the only reason we could probably go back is if we had any kind of COVID research going on. And so the story goes, I started reading about COVID, and I'm going to go quickly over this because I feel like everybody knows this at this point if you've been reading about science. But a viral particle of COVID approaches the cell, interacts with a receptor known as ACE2, fuses, and that the fusion between the ACE2 receptor and the spike protein allows the virus to gain access to the intracellular space. And this releases viral RNA, which is translated by polyproteins, polymerized by RNA polymerase, and this can replicate all of the fragments needed, which is assembled and maturated in the Golgi, and you have a new copy of the virus. So there's 2 parts here that were interesting to us and known in the literature. Disrupting this interaction of the spike protein. This is how most of the antibodies work if you were ever to be hospitalized. Somehow these antibodies are stuck to these receptors and do not allow the spike protein to interact. And then you also have the main protease, which is really important in enzymatically calling these polyproteins, which without them, you cannot really generate or replicate the viral genome. And so we wanted to see if we could build a library around this. There was definitely some things in the literature that allowed us to think about this way. So there was one paper that did a virtual screening of marine compounds and find some polyphenol compounds that interacted with the main protease of SARS-CoV-2. And this came from brown algae. And there was a second study that came out where there was these polyproteins, kind of polypeptidic small molecules. And when I saw these molecules that were also allowed to inhibit the main protease, and there was good understanding of the residues inside the protease that were interacting with these molecules, the first thing I thought about was cyanobacteria. And cyanobacteria tend to produce NRPS, PKS hybrid molecules, so polyprotein molecules. And so this gave me a good understanding that maybe some cyanobacteria could have some effects. So I did, there were some desperate measures here. We bought a kayak. We went out and kayaked. This was, let's say, one of our kayak trips. So we parked here, kayaked for about 6 to 8 kilometers in total this day to collect samples. And I'll come back to why that kayak trip is so important. But in general, this allowed us to go into the ocean. Gracie was a huge driver of this project, and I couldn't thank her enough for being willing to go out in a kayak and scuba diving and snorkeling with me to collect these samples. And so we went around all Puerto Rico, collected 86 samples from 36 sites. At this point, this number has grown. This was only during the pandemic. I think we're about to 125 samples. And we collect the samples. We give them really interesting collection codes, but we also give them really fun names. So this one's Golden Green Harry. And we take those and freeze them, lyophilize them, pulverize them, and vacuum seal them. And then these are ready for extraction. And in this case, we had a really nice collaboration with the Wistar Institute, which we still collaborate with. We're now working on an HIV project for latency reversal agents. But at this point, they had 2 assays up and running, one that can measure the disruption of the ACE2 spike protein through a FRET assay and a high throughput enzymatic assay that measured the ability of the main protease to cleave certain proteins. And so we also, as I said, use metabolomics. So if anybody knows anything about this, we use a mass spectrometer coupled to a liquid chromatography instrument. And so that separates based on retention time. With your MS1, which would be the identity of your compound based on the weight, we can generate compound activity maps using different platforms. And with the MS2, we can use a statistical analysis such as NINDS plots or PCA plots, or we can also use MS molecular networks. There might be some of you familiar with these. And so what we did was use a new tool. In this case, we used a tool called MPACT. And one of the big problems that we have in metabolomics is that if you are to shoot a sample on the mass spec, 30% of what you're shooting on there, and in this case specifically published by the MPACT tool, is that 30% is in your blank. So the things that your data is including is not necessarily things that are in your sample. There's things that are in a very small amount mispicked. You have non-reproducible and in-source ion, meaning things that are colliding inside the mass spectrometer and producing something that's an artifact. And in this case, they were able to establish that in their case, in microbial culture, only about 20% they found were high quality data within their data set. So this is really important for anybody who's doing metabolomics, but for us, this was especially important because we want to get to this high quality data and leave this 80% behind so that our data analysis is much better. And so just as an example, if you're interested, here's the citation for this tool called MPACT, published by the Balloonist Group. If you do no filtering, no impact, just a general statistical analysis, this is what you get. This is your volcano plot. You have a huge amount of differentiation between even just a single biological sample, even in technical triplicates. But then once you filter, you can start to distinguish slight differences. You can reduce the amount of metabolites you're looking at and thus simplify your life. And so I will skip over these slides quickly so we can get to the data. So we do this data, in our case, from our extracts, only about 2.2% is actually high quality data. So we get to filter out a good amount. And what this allows us to do is now generate a nice map of about 18 extracts that were, or 16 extracts that had biological activity against either disruption of the spike protein or main protease of SARS-CoV-2. And they can give us differences between these samples. And something that was interesting to us is that we saw all of these samples all from the same bay of Bahia de Lobos clustered together, and also provide similar biological profiles. And so this was interesting to Marie at the time. And so we underwent kind of a more in-depth understanding, and you can do this by different methods. Here you have like a hierarchical clustering analysis, and you can see that all of these samples kind of cluster in the same clade. And we can also use Pearsman correlation plots, and we can also see that there's high correlation. Here you can see the heat map. The closer you get to this beige color, the more correlated they are, and you can see that, again, we see this correlation. So we were quite confident that these were probably very similar samples in terms of metabolomics, and we decided to investigate a little bit more about their genomic origin. And so here's SAJ34, 44, and 69 as they look in the environment. And we can look at now their microscope pictures, and they're actually quite similar. And we can also then see that their genetic analysis, even though different distributions of this assembly of this Formidia and Oscillatorialis, that they kind of have the same signature, SAJ44 having more of this Formidia. And so when Marie went to isolate the compound, I would expect a compound like an NMR like this, quite complicated, a really interesting structure. What was interested, and at this point this is accepted, sorry, these are some old slides. She isolated a compound like this. And so this was not very interesting for us at the time, but when I looked a little bit closer at the NMR, it's quite easy to solve. We have this fatty acid, which at this point we've called hobosic acid. And it's quite easy to correlate these through COSI HMBC. And then we also did 1-1 adequate experiments to confirm that this was actually the relationship of the two methyl groups. And so just to confirm that these compounds were also present in the other two samples, because we only used SAJ44 for the isolation, Marie conducted some GCMS work, and she was able to detect the compounds quite easily through GCMS work. And something that was interesting to us was just the pure amount of this. So out of 1.42 grams of accrued extract, we were able to isolate almost 20 milligrams of hobosic acid. And then when we scaled up the chemistry, we were able to isolate almost half a gram of the compounds. And so to this day, we don't understand the ecological role of the production of this compound, but for some reason it's there. And what was exciting is that we can follow the bioactivity of the compound is quite active against the spike protein, but not active against a host target homolog, in this case PD1, PDL1, and is also active against the main protease at modest micromolar levels. What's interesting though is that we see no inhibition of other kind of proteases, human proteases that are important, for example, cathepsinol and thrombin. The other thing that Marie did was then tag these, or let's say semi-synthetically access the methyl ester and the phenyl ester. Given that this did not have any other functional groups, we were interested to see if the carboxylic acid was important. And as soon as you do that, we just abolish all kind of activity in this. So it seems like the fatty acid, the acid portion is quite important to the binding, and we still don't really understand the mechanism of action of this compound. The last thing that was interesting, to move on to the next part of my talk, is that one thing that was remarkable is that if you take the wild type SARS-CoV-2, beta, delta, and Omicron, we might remember Omicron because it was quite evasive from standard antibodies and quite contagious, is that your antibodies that are therapeutically available lose efficacy as they get to Omicron. So that you can see here some nice IC50 values for wild type beta, delta, and lambda. Yet when we move to Omicron, we see basically no efficacy. But our compound hobosic acid retains the efficacy. So it does lose some activity, but not in the same extent as the therapeutic antibodies. And so this was quite interesting that somehow, however this compound was probably working in a binding site that was different than traditional antibodies. And so I'm going to jump over future conclusions here quite quickly. So we were able to create a library using metabolomics analysis to prioritize environmental samples of cyanobacteria. We were able to isolate a compound and identify it as an inhibitor or dual inhibitor of both the main protease and the spike protein interaction of SARS-CoV-2. And so lastly, I'll go into one of the newer projects that we have. And I think for all of us, we're interested in how can we find more drug-like and natural product-like chemistry. And we're discovering natural products at an all-time high, so about 1,500 a year. But the novelty of these structures here measured by the percent of these with a Tanimoto score of 0.4 or less is going down. And so Tanimoto scoring is a way of overlapping molecules and determining similarity. And so the lower the Tanimoto coefficient, the less similar compounds are. On the other hand, if you look at natural products, and this is a PCA plot of about 16 physiochemical parameters of compounds, you can see that they're quite diverse in terms of their properties. Natural product-derived drugs are diverse as well, but as soon as you start getting into synthetic compounds that even are natural product-inspired, and then completely synthetic drugs, the space and the diversity in terms of the profiles of these compounds gets quite compact. So there's not a lot of diversity here compared to natural products. And so we had a very basic question, which was, can we design libraries that are easily accessible by chemical synthesis, so synthetically tractable, incorporate natural product scaffolds, but are also drug-like? And so in the interest of time, what we looked at is pseudo-natural products. So if you look at natural products and you break these as if they were Legos, you can deconstruct these and produce natural product fragments and then reattach them in ways that are inaccessible through biosynthetic machinery to produce these pseudo-natural products. And there's a bunch of paper from a group in Germany, from the Waldman group, and we got kind of curious about this in terms of marine natural products, especially cyanobacterial natural products. And so I'll go quick, I'll kind of jump over a little bit here and go straight to bacterial natural products. So what's interesting about cyanobacterial natural products is that they recombine peptide and PKS, so polyketide fragments. And so you have compounds that have either an amino acid that's been cyclized with different kinds of extensions. You have polypeptides that then have extensions. And to me, it's really remarkable that cyanobacteria can be very much like med chemists. So they'll add propionate or acetate functions to create lipophilicity or extensions. They can create amino acids to create selectivity to peptides. And so they're quite nice in terms of the chemistry they can access from very simple and let's say, retained building blocks. And so we started thinking, can we think about this in a different way that we can then design pseudo-natural products from cyanobacteria? So we took cyanobacteria, we divided, we tried to find what was the pharmacophore or what we thought to be an interesting function or part of the molecule and generate the fragments just as the pseudo-natural product fragments. But instead of reattaching them to themselves, we thought to use privileged scaffolds from a variety of natural products. And here you can see those privileged scaffolds that are kind of sustained and very common in natural product scaffolds, not from cyanobacteria. And after we did that, we figured out that if we used amino portions and carboxylic acid portions of the privileged scaffolds, that we could then design the pseudo-natural products through a peptide coupling reaction. In this case, we used a computational tool called Design Hub. We then were able to generate that library in silico, predict physical chemical properties, to lead likeness analysis, and then compare it to drug bank and other databases to see what these compounds would be like. And so quickly, we generate about more than 2,000 compounds. And what we're really excited about is that we have a lot of compounds that obey Lipinski rule of 5, which is a decent indicator for bioavailability through oral administration. And then finally, we have a good amount of compounds that fall into lead-like space. And here you can see the colors 0 through 6 are penalties associated, So these compounds, in terms of functional groups or other kinds of structural descriptors that provide an understanding of how good this will be in terms of being a drug or lead-like. And so we can see a good amount of these compounds are lead-like or don't have any penalties or few penalties. And then also the shape of these molecules tend to be rod-like, but we also have a good spread in terms of diversity. We compare these to natural product libraries in terms of the natural product likeness. I'll draw your attention to two of these curves, one of them being this green one right here, which is synthetic compounds, and then all of these curves on the right side, which are natural products. And ours, the in silico library of cyano-pseudonatural products, kind of occupies this valley in between and also overlaps quite nicely with FDA drugs. And so this is quite nice in terms of the properties that these compounds probably will have. And finally, we've done some comparison with FDA-approved drugs, natural product databases, synthetics, and our pseudo-natural products tend to be overlapping with FDA drug space and natural products, but occupying kind of a new space in between synthetics and these approved drugs. Finally, Gabriel, who's been doing a lot of this computational work, was able to do Tanimoto scoring for all of the compounds compared to different libraries. And this was a huge task because some of these libraries have 2,000 to 3,000 compounds, but some of these have over 1 million compounds. And so what we have here is violin plots of the max Tanimoto score, meaning if I compare each compound of our library to each compound of these libraries, now provide me the maximum Tanimoto score, not the minimal one. And then please provide some kind of statistical information about how many of these there are across the spectrum. So here you can see there's a nice little belly around, let's say, 0.55, whereas we have some other bellies in synthetic products that are higher up into like 0.6, maybe 0.65. But then we looked at the natural products, and when we compare against certain natural product databases, we have Tanimoto scores that are at the very least in the range of, let's say, 0.5 to 0.6. So we are getting a good amount of structural novelty in terms of the compounds we are producing by this in silico method. So I will jump out my conclusion and future directions and just go straight to acknowledgements. We have 5 minutes left, and I want to leave some time for questions. So here's my group right now. Some new members, Miguel and Michelle, just joined last year. If you want to follow us, we are on Instagram. We are soon to leave Twitter, probably migrating somewhere else, but you can follow us also on our website. I would like to acknowledge a lot of the collaborators, Ian, TJ, and Troy, and Joel at the Wistar Institute, some other collaborators from some NIH-funded projects. Here's most of my funding. And finally, I would like to, for all of you who are thinking about graduate school, and I don't mean to steal students from Brazil, but please consider if you would like to live in Puerto Rico somewhere tropical, doing pharmaceutical science research. We have a new PhD program inside the School of Pharmacy at UPR that has three tracks: medicinal chemistry and pharmacognosy, molecular pharmacology and pharmacogenetics, and pharmaceutics and drug delivery. And so if this is something that's interesting to you, feel free to reach out to me through email, scan this code. If it doesn't work, reach through me, send me an email, I'll get that information to you. And with that, I'll leave you with a beautiful picture of Playa Manzanilla and Culebra, and I'll be happy to answer any questions.
Very nice, very nice. Thank you so much, Eduardo. Very, very exciting, very excellent talk about marine natural products. Certainly, all people here enjoy it a lot. Your lecture, and we have some questions about the students here, and let me see. I am trying to translate for you. In English, Esther Cordeiro asked, she said, great lecture. I would like to ask if have some compound from marine life that act to endocannabinoid system.
That's a great. I'm sure there has to be. That I know of, I'm not sure, right? I think a lot of the endocannabinoid system was discovered because of cannabinoids, right? So I think what's really interesting about natural products that a lot of people don't give credit to natural products is how many receptors are named after natural products, right? You look at like cholinergic receptors, yeah, obviously choline, acetylcholine, but there's other of these receptors like muscarinic receptors, right? Inside the cholinergic system come from muscarine, which comes from Amanita muscaria, right? And so I think even it's attest to the endocannabinoids is like, oh yeah, because we knew about cannabinoids, found compounds that acted on the receptors we found because of these cannabinoids, and then called them endocannabinoids. But that's a really interesting question. I'd be happy to look at the literature and see if people have been looking at modulation of the endocannabinoid system. But I'm sure there's a plethora of compounds in plants that do this. And I think that's maybe people have been looking at the marine life for that. But I wonder what would be the function. A lot of these cannabinoids are UV protectants, right? Or they're like these very sticky substances to grab onto pollen. So I wonder if there's a use for sticky substances underwater, especially since these compounds I would expect wouldn't be soluble.
Nice, great. And the next question. Professor Carla wrote, excellent lecture, Professor. My question is for the students who are watching this lecture. What advice would you give to students to want to pursue this area and are just starting out in research?
I would say get research experience in our organic chemistry lab. A lot of the students who join me come to me thinking that we're going to be in the ocean, every day. And I think it's quite the opposite. We do a lot of, round bottom flask extractions using a rotovap, using an HPLC machine, understanding chromatography, right? So I think there's a lot of literature out there. And I think I would really tell students, I grew up in this day and age where you can go on YouTube and there's channels full of information, and there's people on Instagram that have full profiles dedicated to synthesis of organic compounds or natural products, right? But that would be my biggest suggestion, is to get really good at organic chemistry. Like we use a lot of NMR, we use a lot of chromatography. And for the medicinal chemistry part, I think that's much more teachable, right? Because usually we don't do a lot of the assays ourselves, and collaborations are, even with UFIs, we've collaborated because maybe there's a new assay system that we want to look at the natural products. So it's going to depend on what side of the natural products you want to look like. Mm-hmm. So if they want to look at like isolating compounds, go for organic chemistry, try to get some good fundamental training in that. If they want to look more at the medicinal chemistry, chemical biology, what are some cool new receptors out there? Read in the literature of what therapeutics we need. I think right now one of the biggest needs we're going to have is pain, right? Especially with pain is going to be a big area for the next 20, 25 years, because we're trying to move away from opioids. And I think the other will be anxiolytics. I think we're going to move into a world where we're going to have a lot of anxiety. There's a lot of changes in the world, and we need safer anxiety-reducing medications. Because I think cancer and some of these other diseases, they have some good drugs out there, and there's a lot of the antibody drug conjugates are there. So I guess that would be my suggestion. Get into a lab, get your hands onto glassware, do work, and see how you like it.
Yes. Jéssica Monteiro, Jéssica, I would like to ask if the works about the library are available in the article format.
So soon, we're actually almost publishing the work on the pseudo-natural products. And so one of the things that came out of that work that Gabriel did a fantastic job was to curate the databases. So there's like Coconut 3.0, there's like all these databases of natural products. And when we started looking through the structures just manually, we found a lot of synthetic compounds in there, or compounds that didn't belong. They were just, people include these huge libraries, and at some point, how do you curate them? So as soon as we publish those, the libraries we use for comparison will be as SDF files in those publications. And we hope that those data don't require, access through a paywall. That, usually supplemental information and files are free. So we definitely can do that. So hopefully the library now will be small, but we're hoping soon to use some AI fragmentation tools to generate libraries of 500,000 compounds and then really dive deep. And so there's some future work there. So as soon as we have that, we'll be happy to share it and maybe get the publication around to everyone.
Yes. One more. Rita asked, how do you choose the location here where you collect marine samples?
That's a great question. I don't have a good answer for it. I think we do Google Maps. So we look at Google Maps, we see like, oh, this little corner looks interesting. It goes from like, okay, so why? Okay, let's go check it out and see if we can see anything. And then we document a lot of it in our notes of what kind of organisms we saw, what the temperature of the water kind of was, what was the overall feel. I think the more and more, my suggestion to people at the beginning of collecting is just go collect everywhere that makes sense and is easiest. And as you start learning, try to look for patterns. At what depth were we? Was there a river close by? These kind of things can change, but you can make notes and then you can make good predictions. And I think we've gotten to the point where we make a prediction, we pick a point in the map, and we go and we find stuff.
Yes. And have you been using artificial intelligence or not?
So that's the next step in this. So we want to do some AI docking, and we also want to do AI fragmentation generation. So one thing we got really excited about is that there's a lot of people thinking about fragmenting libraries to make, fragment-based libraries, which, by the way, is another beautiful way to do medicinal chemistry. You can start docking small little bits of fragments of things and then start putting the molecule together and then generate your molecule, which is something we thought about. So now we're thinking about, can we use some of these tools, feed it a natural product library we've curated, generate fragments that are make sense, right? Through very detailed fragmentation rules. And then that fragment library, feed it into a second tool that will then make the pseudo-natural products. We create a big library. And so like a lot of this is ongoing work that we're presenting.
Is the answer of $1 million. Rita wrote. Mauricio Mata, how are you preparing in the face of climate change?
This is a good one. I think for us, we're going to be able to see the impact in the environment firsthand, right? I think the organisms that are going to be there are not going to be there anymore. Some new organisms will move in. So I think there will continue to be opportunity. I think the question has a political side, which is living on an island. How are we preparing in the face of climate change? And I want to say at a Puerto Rico level, I don't think we're doing a very good job. So I think there's definitely there's space for improvement in terms of really taking seriously how these coastal environments are going to change, how we are going to be impacted by those changes, but also how we're going to more closely impact the coast because it's going to be kind of closer to us. And so I think there's a lot of questions there. Mauricio, I'll tell you in about 5-10 years how we're surviving.
Yes. One more? No? Eduardo, thank you so much for a lecture, for your time. And I think, I certainly that people enjoyed a lot your lecture. We learned a lot as well. And very good.
Thank you for having me. And I hope this inspires those students to pursue natural products. There's so many ways you can look at these. If you're interested in medicinal chemistry, there's places for you. If you're interested in chemical biology, in chemical ecology, in metabolomics, if you like more computers and workflows and coding, there's a lot of work to be done in this field and a lot of new opportunities.
Nice. So, vamos dar continuidade, só palavras finais, né? Eduardo, muito obrigado novamente. Vou passar a palavra para Tatiane, que é pós-doutoranda aqui do Programa de Pós-Graduação em Ciências Farmacêuticas. Ela vai, né, passar algumas informações finais, né, e a continuidade do workshop na segunda-feira. Então, boa noite a todos.
Obrigada, professor Rodrigo. Boa noite a todos. Queremos agradecer pela presença de todos, pelo professor Eduardo ter aceitado o convite, que foi gentilmente realizado aí por meio da professora Rita. Agradecer, aliás, a todos os professores que sempre nos apoiam, o professor Rodrigo aqui nos apoiando na transmissão, os professores do programa, no chat, nos ajudando aí também, nos apoiando. Os alunos do programa de pós-graduação em ciências farmacêuticas que estão envolvidos com este evento, nosso muito obrigada. Bom, essa foi uma palestra do nosso workshop. O workshop é um evento que nós realizamos anualmente para apresentar as linhas de pesquisa do programa de ciências farmacêuticas da UFES. Nós temos mestrado e doutorado. Inclusive, o edital de seleção do mestrado está aberto. Essas informações vocês encontram lá no nosso Instagram, @ppgcefa.ufes. Lá tem essas informações, tem o link do formulário de presença desta palestra para posteriormente podermos enviar o certificado de participação. Tem até a presença do professor Eduardo, quando ele esteve aqui conosco, tem alguns registros lá. Tem muita coisa interessante, bacana, então siga nosso Instagram para vocês ficarem por dentro de tudo. Esse formato online aí, principalmente herdado da pandemia, foi uma adaptação que nós fizemos no nosso workshop, então a parte online, que foi hoje, a palestra, a parte presencial será na segunda-feira, e é interessante porque aproxima, né, nos aproxima de pesquisadores. Então a gente sempre vai trazer aqui pesquisadores do nosso estado, do nosso país, e pesquisadores estrangeiros, como foi o que nós trouxemos hoje. Então, essas palestras de abertura também são fontes de informação, de novidades, então é muito importante. Agradecemos a todos, aos inscritos no workshop. Até segunda-feira, nos vemos lá presencialmente, e a todos os demais aqui presentes, obrigada, uma boa noite, um bom descanso a todos, e nos vemos no próximo ano, com mais uma palestra, com mais um palestrante, um pesquisador aqui, trazendo novidades, temas importantes na área da pesquisa. Até a próxima, uma boa noite a todos, tchau, tchau.